The main objective of Work Package 7 is to develop tomato lines that have improved food quality traits, mainly flavor and health-related compound composition, while maintaining characteristics of interest for producers and other stakeholders, such as productivity, shelf life, and good performance under different conditions. 4 tasks were performed or are underway. In the first task, CSIC and INRAE analyzed a total of 32 studies from the research performed in different countries. For each trait related to food quality, the most favorable QTL regions, alleles, markers, and genetic resources carrying them were identified. Data were organized and provided to Work Package 3 to be entered in the Harnestone database. UNINA selected 14 studies analyzing tomato volatiles together with flavor and aroma, and they identified about 100 volatiles which are important contributors to tomato aroma. The second task consists in improving sweetness, acidity, and volatile composition by the introgression of QTLs into elite materials. Based on the information from the previous task, pre-breeding lines are developed. In CSIC, they started, for instance, by validating and fine mapping 15 QTLs carried in 6 introgressed regions, and they tested their impact through sensory analysis. This year, they focused on the 2 most interesting regions and started characterizing the QTLs. The other partners, INRAE, Gauthier Semences, and Villemorin, started marker-assisted selection after selecting different elite materials and donor lines and breeding regions. They performed several backcrosses and used marker-assisted selection on hundreds of plants. In parallel, UNINA used new protocols to assess the volatile profiles of breeding accessions of semi-ortho and correlate the volatiles with sensory data. These materials will be tested by sensory analysis and volatile content next year. In the third task, ENEA and INRAE are presently studying 2 F2 populations for shelf life and health-related antioxidants, and QTLs will be mapped from this experiment. In the fourth task, we are optimizing pre-breeding methods for several quality traits simultaneously. A population for the long-term improvement of several quality traits is currently under construction by Villemorin and INRAE. It will be studied next year for fruit quality traits and genome analysis. INRAE is also analyzing the most appropriate genomic prediction models in order to predict the best performing accessions. Finally, the original materials and the introgressed ones will be assessed in different growth conditions next year in collaboration with CTAX. Thus, this Work Package 7 will provide new information, but also molecular markers, protocols, and plant materials to breeders for the future creation of tomato varieties with improved fruit quality. This should also, at the end, satisfy tomato consumers.